just wandered in from the Insectarian butterfly house. Thank you so much for joining us this evening. My name is Brenna Shea, for those of you I don't know, and I am the development director and one of the co-founders of the Butterfly House. As spring has kind of settled in over these last couple of months, you've likely noticed with excitement like me the annual return of bugs after a mostly bugless winter. But with the long daylight hours and the warmer weather finally arriving, it also signals the annual Bugs and Brews summer hibernation. So tonight's going to be the last lecture of the 2024-25 Bugs and Brews lecture series, and I hope you've been able to join us for a number of lectures over the past, we started last September, and have learned so much more information that you are now storing in that buggy part of the brain. We look forward to kicking the 25-26 lecture series off again in late September. And I'm really glad that we are taking a short break, because if a lot of you know the tradition that we have when we kick this off, this is going to give me a little time to add to my list of jokes, because the well is getting a little dry, and so here are 2 fresh jokes for this evening. And I'll say they're fresh jokes, not necessarily good jokes. Okay. So Bob and Jane went out for a really romantic dinner. During their conversations, Jane challenged him to undress her with just his words later that evening. So after returning home, entering the bedroom, dimming the lights, he suddenly turned to her and said, "I just saw a spider go down your shirt." Good, that was better than I thought. All right. This one's just a cute one. What do you call it when you have lots of spiders in your house? A no-fly zone. Okay, well, just so, because we've got this seasonal change has signaled the dormant period for Bugs and Brews, it doesn't mean that you can't go out and enjoy a delicious DraftWorks beverage out on their patio. DraftWorks has been an incredible partner for us for a very long time, and if you haven't had the chance to have a relaxing evening out on their patio sipping a beer or a hard seltzer, you should definitely check it out. They have been, for the last 8 Bugs and Brews, they've donated all the beer, all the hard seltzer for all of the lectures for the 2024 and 25 lecture series. Of course, that's very generous, but that's what they've been doing since 2015. They've been, again, incredible partners. It adds up to a lot of buggy knowledge being shared in a fun and really relaxed environment. So thank you to DraftWorks for putting the beers in the Bugs and Brews. Okay. All right, so since we're taking a little bit of a break, I just want to take a moment to just say thanks to a couple groups of people. First off, our staff. And, all of our staff are here from time to time for Bugs and Brews. But Carolyn, I don't know where she is. She's probably doing something right at the moment. But Carolyn Tabor is up here all the time. Jan Marangelo, my lovely wife, and as well as, and Stacy. I'm not sure where Stacy is, but Stacy's around here too. But, and then aside from them, the rest of our staff, we really appreciate their assistance. All of our volunteers, so a lot of the people that were wearing the orange vests, handling everything from crowd control to showing off some of our bugs at the encounter area, and bartending. We've got this crew of bartenders that pretty much been with us almost every Bugs and Brews for a long time. So we really appreciate your time and your talent and just for sharing yourself with us. Our friends And then new since January, a huge thanks again to Missoula Community Access Television, better known as MCAT. This is the 5th lecture that they're recording for us as part of a media assistance grant donated to the Missoula Butterfly House and Insectarian. And I'm happy to announce that we're currently in the process of hiring a new marketing and events coordinator. And once this position is filled and we turn a little bit more attention to the maintenance of our website, which we hope to include a section on the website where you'll be able to go back and check out some of the past lectures. We've only had 5, but we hope to continue this moving into the future because we always have some great speakers. One little thing of housekeeping. Instead of just telling you what events are coming up, I'm just going to mention this one because it's kind of a new thing that we're trying. But on June 17th from 5:30 p.m. to 7, we're going to be having a butterfly photography class or workshop. I'm not sure exactly what we're calling it. But if anybody knows Kathy Eister, she's been teaching photography courses for about 25 years throughout Missoula. Teaching at the Lifelong Learning Center, the Rocky Mountain School of Photography, and also through private instruction. And so she's specifically, shares some of the tips that she has on butterfly photography, and then you'll be able to go and practice that in the butterfly house and see how well you do. This is open to anybody with any kind of camera, from the big setups to a smartphone. And normally tripods and monopods are not allowed in the butterfly house, but of course for this we're going to make an exception. So again, that's Tuesday, June 17th, 5:30 to 7. If you remember, it's $35. If you're a non-member, it is $45. And so we're excited to have that, because I know we are always trying to get some great photos of not just the ones in the butterfly house. It's more challenging to get the ones outdoors sometimes. So for tonight, I'm really excited for tonight's presentation. It comes as a little bit of a mixed bag because we are welcoming Brenna Shea as our speaker. Most of you probably know her better as Bug Wrangler Brenna. I just get tongue-tied on the Wrangler part. But at the same time, we're saying goodbye. So after 6.5 years of sharing the amazing world of insects with learners of all ages through a variety of education programs, Brenna has moved on to the next chapter in her life regarding her career. But over these 6.5 years, she actually had started as a volunteer years ago, came on as part of our animal care team, and while she handled a lot of different important tasks, it wasn't until COVID hit and we launched our online bug encounters that Brenna really found her calling as Bug Wrangler Brenna. In addition to helping with a variety of other education programs, as it gives a writer and an informative and really comfortable style in front of the camera or an audience, and much of her recordings were audio, so that was always impressive. But Brenna developed a weekly following of bug lovers for the encounters. And with a quickly growing base of knowledge of insects and arthropods of all kinds, in addition to online bug encounters, Brenna took over putting together our seasonal What's Buzzing posts, wrote our notes from the lab reports, and she also took on several community science projects that we were just getting started. From her personal experiences with many of the live animals in our collections to her research on many bizarre species and behaviors, that Brenna is certainly someone well-versed in regard to some of the weirdest and wildest insects around. Brenna grew up in Western Washington and moved to Missoula in 2013, and growing up in a rural part of the Pacific Northwest was a really kind of a formative place to grow up for somebody in terms of developing a love for the natural world. It led to many outdoor pursuits as an adult, and after finishing a stint with the Montana Conservation Corps, Brenna completed a Bachelor of Science degree in biology with a concentration in organismal biology and ecology. While this may be the last time we introduce her as Bug Wrangler Brenna, tonight I'm thrilled to introduce Brenna Shea as she takes you on a tour of all the different things she's learned her journey through at the Missoula Butterfly House and Insectarian, introducing you to some of her favorite insects and topics.
All right. Can you guys all hear me okay? Yeah? Okay. Awesome. Well, as Glenn said, my name is Brenna. I see a lot of really familiar faces in this crowd, including many friends of mine who have already been held captive to a lot of different bug rants. So I really appreciate you all being here. to listen to this other one. I love having a captive audience for a solid 45 minutes because we're going to go down a road of really, really weird bug topics, and I'm really excited to take you all with me. So as Glenn mentioned, I have been with the Butterfly House for about 6.5 years, or was. I have recently moved on to another job, but the knowledge and experience that I gained while I worked here at this organization, it really just cannot be understated. I've loved my time here. I assume a lot of you have seen the online bug encounters and read notes from the lab. If you haven't, I won't hold it against you, but they're really good. But a lot of that knowledge was not knowledge that I came into this organization with. I learned everything here. When I first started, I had no idea about bugs. In fact, I didn't really care about them that much. So today I really want to take you through kind of like what my journey at the Butterfly House has looked like and some of my favorite topics that I have learned along the way. So when I first started, this organization looked a lot different. Does anyone recognize this photo and where this is? Yeah. So this was our Front Street location in downtown Missoula. This is where I came on board. This is where I became familiar with this organization. It was a very small space. What you see there is most of it. There's not really anything too much more on that other side of that other wall there. But this is where I got my start as a volunteer in 2017. This is one of my favorite pictures. This was my first volunteer event. This was Halloween 2017. It was probably the first time I ever held a bug, and also one of the first times that I ever really interacted with children, if you can't tell. So I got my start as a volunteer, and while I was volunteering, I was also working towards a bachelor's degree in biology. As I mentioned, I didn't know a single thing about bugs when I first started. And I think it's important to note that I didn't go to school until I was about 25. I didn't go to college right after high school. I'm a millennial, so I'm a part of that generation that was told, like, college is the path. And I graduated from high school, and I was like, well, yeah, but, like, I don't know what I want to study. And I've always been interested in science and biology, but to be honest, when I graduated high school, I just didn't think I was smart enough. I did not think I was smart enough to pursue a biology degree. It sounded so intimidating. You have organic chemistry, you have physics. I was like, there's no way. I have really smart friends who couldn't handle that, and if they couldn't do it, there's no way that I could. But after living in Missoula for a while and kind of stagnating in the customer service industry, please tip your baristas and your bartenders. Like, please. I decided, like, why not? I'm not doing anything else. I might as well give it a shot. And that was where I kind of became familiar with the Butterfly House and Insectarian, was while I was in college. And I really decided that it was worthwhile to kind of immerse myself in this volunteer gig. It's like, I really enjoy this. I don't know what I'm talking about, though. And entomology was a part of my academic track, and so I figured, the timing seems to work out. I might as well give entomology a shot and try to learn a little bit more about this organization that I'm trying to support. I really wanted to be able to engage with visitors and have a conversation and provide them with information that they can, solidly know was true. When I first started, there's one particular instance, and I don't think I've ever told Jen or Glenn this, so I'm sorry. But I did what we tell all volunteers they shouldn't do. And when a guest asked me a question, I guessed the answer. So if you are familiar with our Front Street location and have been familiar with the organization for a while, you're probably familiar with Polly. She was our Goliath bird-eating tarantula. She has sadly since passed on. But she was enormous, and she was really mean. And at one point, when I was volunteering, I was sitting at a table kind of across the museum, and there was a visitor. He was the only one in the museum at the time that was looking at Polly and her enclosure, and there was a little exhibit card down here that he just didn't see. And he kind of called to me from across the museum, and he said, "Wow, the spider's really impressive. Where is she from?" And I was like, "Oh, I don't know. She's really big. Australia, probably." So I got up and I walked across the room, and then I looked at the exhibit card and saw South America. And I was like, "Oh, wow, really? That's insane." But I mean, like, look at her. Would you not assume that she was from Australia? Like, she's huge. So that was kind of my moment where I was like, I should probably educate myself a little bit more on the subject before I try to talk to museum guests. So today, we're going to talk about kind of 3 big topics, as long as we have time. We might only get to 2. I like to talk about bugs if you... I'll try to get to 3, I promise. So we're going to talk about 3 big topics. I want to talk about parasitoid wasps, a topic that is very near and dear to my heart. I want to talk about flies because they are a really, really cool group of insects that are very underappreciated. And I want to talk a little bit about DNA barcoding and eDNA and some of the projects that we, and the community science projects that we have incorporated into the programming at the Butterfly House. So hopefully we'll get to all 3. I'll try to keep it somewhat brief. So, actually, before I move on, I would like to say, some of these topics will be a little gross. I'm just telling you now. When we talk about flies, one of my favorite things to talk about in terms of flies and their life history is how humans can utilize them in scientific applications, including medical science, i.e. medical maggots, and forensic entomology. I'm also a very anxious person, and the idea of being shown a bunch of really gross imagery really stresses me out. So I'm going to make you a promise, just like I like to make the kids promises, or did when I was still educating here. I would always promise the kids, if you don't want to hold a bug, you don't have to. And I'm going to promise all of you here right now, I'm not going to show you images of like gross necrotic wounds and corpses, okay? Just want to put that out there. But that said, we are going to talk about some really gross topics, and there is some really gross imagery, just not that gross imagery. So I want to start with parasitoid wasps. I want you all to picture a wasp. Picture a wasp in your mind. I'm going to go ahead and guess that it probably looks like this. Yeah? Yeah. So this is a yellow jacket. Obviously, we're all very familiar with them. This is not a good representation of what wasps in general look like. Wasps are incredibly diverse. Some would argue they are the most diverse, but we'll get to that in a minute. So, parasitoid wasps in particular range from some of the smallest species of insects that we know to about 1.5 inches long, i.e. the tarantula hawk wasp, which I think a lot of people are familiar with. Most females have this really long ovipositor. This is not a stinger, and that is what she uses to lay her eggs. So, all wasps, including parasitoid wasps, belong to the order Hymenoptera, which also includes bees and ants. It contains about 150,000 known species. And to put that into perspective for you, the subphylum that vertebrates belong to contains about 65,000 species. So vertebrates, that's anything with a spine and a central nervous system, right? So pretty much all of the bigger animals that we are familiar with on a day-to-day basis. Mammals, fish, birds, people. But this is just what we know about, okay? Does anyone know, besides the people that work for the butterfly house, what the most speciose group of insects is? Beetles, the Coleoptera. So currently, we know that there are about 400,000 described species of beetles, but most beetle scientists believe that there are well over 1 million. But then if you talk to a hymenopterist, a wasp, bee and ant scientist, they will tell you that no, beetles aren't it, it's wasps. There are way more wasps. They believe that there are several million wasp species, and that's specifically wasps. I'm not talking about bees and ants here. They believe that there are several million species of wasps. And the reason for that is because a lot of the species are parasitoid species, and many of them are tiny, tiny, tiny. Not only that, but they are solitary. So they are really, really hard to study and really, really hard to identify. I always like to joke, a coleopterist and a hymenopterist walk into a bar, but there's no punchline because it's the most boring conversation you've ever listened to, and they're both just fighting over whose animals actually dominate the planet. So this is a very blurry Wikipedia video of a braconid wasp that is one of the groups of parasitoid wasps. This is a female. She's about to find her host. So that's what parasitoid wasps do, is they seek out a host for their offspring. Hopefully, there we go. So a female wasp will seek out a suitable host, and she will lay her eggs either on or in that host for her offspring to feed on. So that's an ant. This particular species target host is an ant. So looking, you all know how big an ant is. And then see the size of that wasp? That's what I mean when I say they're really, really tiny and solitary and hard to study. They are minuscule. So this particular wasp is going to lay her eggs either on or in her target host. So let's talk about that, these different types of parasitoidism. So broadly speaking, there are 2 different types of parasitoidism. We have the ectoparasitoids. Those are the ones that lay their eggs on the external body of their host. This is a tarantula hawk wasp. This is probably the best known example of this. This is also one of those really, really big wasps. And their sting is very painful from what I hear. These guys will paralyze their hosts immediately. So the host is still alive because their offspring don't like to feed on dead tissue. They like to feed on live things. So they paralyze their host and then lay their eggs on it. And then the eggs hatch and they have just a nice meal.
Do they stay paralyzed?
Yes, they are paralyzed throughout the whole process. Which brings up a good point. You'll see me say the word parasitoid, not parasite. And that is because parasitoids, it always ends up in death of the host. Always.
How long does it take them to hatch?
These life cycles are pretty short, so it depends on the species, but I would say probably a week or 2. How long it takes them to hatch, yeah. But my favorite type of parasitoidism, and the one that I'm going to gross you guys out with, are endoparasitoids. And these are parasitoid wasps that lay their eggs inside their host. And they don't paralyze their host because they need their host to live and grow so that those babies inside it can continue living and growing with it. Okay? So, real gross. Let's see here. The most common example of this that we see is in caterpillars. So, I have here, live and grow for now. I forgot I added that. So this is a video of a parasitized caterpillar. And I hope it plays. This is a really gross one. The wasp larvae that exist within this caterpillar have already grown to size. So they are ready to emerge. They want to pupate. They want to grow up into adult wasps. So once they are done feeding on their growing host, they will sort of erupt from the body. Oh no, please play. It should play. Here, let's see if I can do this. I'll just do this. Oh my gosh. It's really gross, I promise. I don't think it's going to load.
A quick question while this is loading.
Please.
So the eggs are already fertilized when they're...
Yes.
When is that?
Mm-hmm. Yep. So the eggs are already fertilized. I don't know if it's going to load. That's so disappointing.
And then, will this kill the caterpillar?
Yes. Once they emerge from the caterpillar, it's all over for that caterpillar. And a lot of times, what they will do is they will emerge from from the host, and then they will spin a cocoon on the outside of the body. So if you've ever seen those pictures of the tobacco hornworms, those really big green fatty caterpillars with little white things sticking out of them, those are wasp babies. Yes. I'm going to hang on to questions until the very, very end. Yeah, oh my gosh. I meant to set a timer for myself because I don't know if you can tell, but I like to talk about bugs, and I can get carried away sometimes. All right, we're not going to wait for this video to load. I'll find a really gross one at the end while we're answering questions. So let's go back. So, big problem when it comes to injecting your host with eggs. Insects have immune systems, just like we do. And immune systems are very, very good at fighting off infection and foreign bodies. And so in order to bypass that immune system, wasps have to do something kind of weird. They have to recruit some help. And they do that with a virus. So this is a really unique example of a mutually beneficial relationship between a wasp and a virus. This is not really something you see in nature. We don't really coexist with viruses very well. I don't know if you guys remember COVID-19. So this is a group of viruses known as polydNA viruses. There are two different types. There is the ichnoviriforms and the brachoviriforms. And this is kind of my favorite part about this virus evolution. I'm not going to get too deep into the weeds, but parasitoidism evolved once within the Hymenopteran lineage, and it's extremely ancient. It was one of their first evolutionary strategies. It's how they proliferated and sort of started dominating the insect world. The wasps and bees and ants that we are familiar with that live in these eusocial communities, so honeybees and yellow jackets and all ants. Eusociality evolved after those groups lost their ability to parasitize other animals. So it evolved once, and then a bunch of insects lost it. But this mutually beneficial relationship with polydNA viruses evolved twice. So this is what we call convergent evolution. And for those of you that are unfamiliar with the term, just think of it in terms of birds and bats. So birds have wings and bats have wings, but they didn't evolve from each other. They're completely different organisms. And that's what happened with poly-DNA viruses. It popped up randomly twice within the evolutionary lineage and then kind of went on their own directions. These viruses are not related whatsoever, which just shows you that it's a very, very beneficial evolutionary strategy. Even Charles Darwin was disgusted by parasitoid wasps. I think we all know who Charles Darwin is. I'm not going to give you a lesson here. But this was kind of the nail in the coffin for him in terms of like, yeah, I don't think God had anything to do with this. Like, this is bad. This is really bad. I cannot persuade myself that a beneficent and omnipotent God would have designedly created the Ichneumonidae, that's one of the parasitoid wasp families, with the express intention of their feeding within the living bodies of caterpillars. So he was like, yeah, I think there's this thing called evolution. I don't think God has anything to do with it. So, all right. So, it's a little palate cleanser, right? So, because that was kind of a gross topic, I really wish that video would have loaded. I think that would have really carried the weight of that conversation a little more. But, so, little bug fact for you guys to take away with you that will hopefully help you dominate trivia night one day. Did you guys know that bugs fart? That's one of my favorite bits to tell kids. You always have that one sassy kid that's like, do you guys have any questions? And they're like, yeah, do bugs fart? And you're like, yeah, actually, they do. All right. Let's move on. I had a video for that too, but we're running out of time. All right, let's move on to flies. Flies are one of my favorite group of insects, and I don't think a lot of people can really say that. So when I started at the Butterfly House, as Glenn mentioned, I came on as an animal care employee, but I also started writing the Notes from the Lab blog. And in the early days, it was just animal profiles of some of the animals in our collection. But we were pretty small. We didn't have a lot of animals at the time. So after a while, I started to explore other topics of interest and started reading research papers, and very quickly became very fascinated with flies. I really like to champion the underdogs. You know, they're kind of the underdogs of the insect world. They're like the pain in everyone's ass. But they are really, really fascinating insects, and, you know, without them, we wouldn't exist. You could say that, I think, about a lot of all insects, really, let's be real. But flies perform so many different ecosystem services that we are totally unaware of. Decomposition is obviously the biggest one that we're all familiar with. But they're also crucial pollinators. They dominate pollinator networks in the Arctic Circle. You're not going to find any other pollinators up there, especially the higher you go. And they also form the base of these really, really crucial food webs. So they're feeding a lot of those larger animals, the trout, the migratory birds, all of it. We also, oh yeah, that's right. The largest living land animal in Antarctica, and exclusively terrestrial. So you think of like penguins, they are semi-aquatic, everything else there is semi-aquatic. The largest terrestrial land animal in Antarctica is a fly. Oh, where'd it go? There it is. The Antarctic midge. There it is. Largest terrestrial animal. So flies also are responsible for pollinating some of our favorite foods. So does anybody know what this is? It's cacao. It's chocolate. So chocolate, cacao trees, are pollinated by the chocolate midge, which is a type of no-see-um. You guys like no-see-ums? No, nobody likes no-see-ums. Without them, though, we wouldn't have chocolate. They are the primary pollinator of this plant. You can see this flower is extremely complex. It grows like straight out of the trunk of the tree. They're really weird looking. They look like they dropped out of a Dr. Seuss book. And these chocolate midges, these tiny little biting no-see-ums, are the only things that can pollinate it. So without them, we wouldn't have chocolate. And I'm not sure that's a trade that I am willing to make. Flies, as well as other insects, are also involved in forensic investigations. So when a victim of a crime is found, one of the first things that investigators want to do is determine a time of death, right? That's pretty crucial to the investigation. And a body will start to decompose pretty rapidly after they die. And almost immediately, insects will move in to help with the decomposition, if you catch my drift. And different species move in at different times, and they also will grow at different rates. So when a forensic investigation kicks off, one of the things the investigators want to look at is the species that exist on that body and what stage of life they are in. And then by kind of backtracking, they can determine when that person or animal, they use this for animal investigations as well, you can determine when they actually died. So they're very useful tools. And again, it's not just flies, but they're probably one of the more famous ones. But my favorite application of flies and their offspring has got to be medical maggots. Yeah. So using maggots, which are fly larvae, as a means to, man, none of these videos are loading, treat infection dates back to antiquity. And this is still something that we use today. You can still get some medical maggots if you have a wound that won't heal. So this is called maggot debridement therapy, or MDT. And there's a lot of evidence of a lot of indigenous groups using it. And the reason this is so useful is because these maggots will consume necrotic flesh. Yum. And they're so precise that they are significantly more efficient than any surgeon, even the most egotistical surgeons. So the larvae, these maggots, consume the necrotic tissue. Usually takes about a day or two, so they're pretty quick. They also, being living organisms, secrete a lot of stuff. But these secretions have been shown to have broad-spectrum antibiotic properties. And research shows that they can destroy a wide range of bacteria, including Staphylococcus aureus, which some of you might know better as MRSA. So, because this process takes a couple of days, and nobody really wants to witness maggots consuming dead flesh in an open wound on their body, the wound is often dressed in a way that will prevent exposure to the elements, but also provide the maggots with enough oxygen to live. They still breathe air. But they're also dressed in a way to kind of minimize the tickling sensation that the maggots cause while they are in the wound. Yep. All right. Palate cleanser. Did you guys know that bumblebees play? Did anybody read this study? This was a fun one. Oh my gosh. So bumblebees, in this study that came out like just a couple of years ago, were shown to play with these little wooden balls that they're rolling around. Oh, the Wi-Fi is really bad in here. That's why. So unfortunately, I don't think you're going to be able to see this very adorable video of this bumblebee playing with these wooden balls, but it was shown that bumblebees will engage in play activity similar to what we recognize as play. I highly encourage anybody to, even if you don't have a scientific background, look up this study. It's a really fun read, and it's really cute. Okay. We are very quickly running out of time, so unfortunately I'm going to skip my last topic, which was about the Citizen DNA Barcoding Project and eDNA. So I'm going to skip through it. I'm going to share one last bug bit, one last little palate cleanser, even though that last topic wasn't really actually that gross. But anybody know what this is? Close. It's a damselfly. They belong to the same order, but they are the most efficient predators on the planet. Dragonflies and damselflies have what a lot of people would refer to as primitive wings. Most insects, when they flap their wings, they're not like actually flapping them. What they're doing is they're compressing their abdomen or their thorax, excuse me. Wow, that's embarrassing. And their wings are attached to thoracic muscles that when the thorax compresses, that will then move the wings. And that's why insect wings all move kind of in unison. But dragonflies and damselflies have very primitive wings where they each have their own individual flight muscles. So they can move each wing independently of each other. And not only that, but they have enormous eyes. And these two things make them really, really efficient predators. I believe they can catch up to 94% of the prey that they pursue, which is unheard of. So, circling back kind of to the beginning, this was like a lot of information to kind of download. And honestly, if you let me talk about everything that I learned in my time at the Butterfly House, it would ruin your Memorial Day weekend plans. And probably cut into your work week next week. Like, I've learned so much during my time here. And I just want to reiterate the point that, like, there was a time that I didn't think that I could do any of this. And so, if you walk away from here and you take anything from this talk, I would love it if you took the bug bits. I would love it if you look into medical maggots a little more. But if there is one thing that you take away from this, I want everyone in this room to remember that you are smart enough and you are capable of doing this, because I am, and I'm a terrible student. So just keep that in mind, and if I can't convince you, then I want you to take some words of wisdom from my very first teacher. She is the person that inspired my love of science, my love of the outdoors, my just overall curiosity. Let's see if we can get this to play. Yes. Let's see if we can get the sound to work. Hold on. Wait, let's run it back one more time.
Speaking of dropping, that's not a bad idea, Ralphie. Take chances. Make mistakes.
Take chances, make mistakes, and get messy. And most importantly, I was just going to keep playing. But most importantly, everyone, please stay buggy. I will take your questions now. Yes. What's that? Oh, I go to like the family level and then I give up. There's a lot of them.
You can't tell anything beyond family without putting it under the microscope.
Exactly. And unfortunately, because we had to skip the last topic, the Citizen DNA Barcoding Project talks a lot about why we can't really rely on classical taxonomy anymore. I mean, we can rely on it. It's a very reliable method. But unfortunately, you have to be an expert. And there are very few of those out there, and it takes a long time to learn. But you are more than welcome to reach out to me personally if you want to learn more about that. Yes?
When the wasps erupt from the caterpillar, is it because they get so big that they burst, or do they chew their way out?
They chew their way out. Yep.
This was also about the wasps. So is the mating like a deposit, like it is with centipedes, where they just like, leave a sperm sac somewhere, or is it like an active mating ritual that doesn't happen in male and female wasps?
That's a good question. I am not totally sure. I believe it's just based on what I know about honeybees, which it's a darn shame that Scott isn't here, our bee guy, because he can provide you all of those mating details, like very well. But I'm pretty sure it's a direct. But the virus itself replicates within the ovary of the female wasp and is passed down to her offspring. So yeah, the males honestly and like the whole parasitoid wasp phenomenon are kind of uninteresting. You know, they don't have ovipositors, they don't have the viruses. They're kind of just there to help make the next generation. So, yes.
Is there any chance you could send out the links to the videos later?
Yes.
You might want to watch them.
Oh, absolutely, especially the bumblebee video. It is really, really cute.
How do the maggots know not to eat live flesh? Do they just not like it?
They don't like it. So when it comes to fly larvae, you know, every species is different. Some species do consume living tissue, but the species that they use for the medical maggot treatment are from blowflies, which are those really shiny metallic green flies that you see, and they only eat dead tissue. Yeah.
Do maggots also secrete like an anesthetic at all, or can you literally feel it?
You can feel it. Yeah. Yeah. Yep.
Are you excited about your future job? Is it with bugs?
It is sort of conservation adjacent. There are conservation elements, but I probably won't be writing about bugs as much as I did with the butterfly house, which, you know, is a shame because they're really fun to write about. But, you know, I might start a blog or something, you know, who knows. Yeah.
What is eDNA and how does it relate?
Oh my gosh, I'm so glad you asked. So eDNA, or environmental DNA, is DNA that we are able to extract from the environment. And it's being used in a lot of really cool ways. To put it very briefly, we are able to isolate DNA from target species, and we don't need to have like a visual check to know that those, like we don't need to collect the species themselves in order to get their DNA. So the first sort of environment that this was used in was in aquatic systems, where someone could take a sample of water and extract the DNA that was present from it and use that DNA present in that water sample to identify the different organisms living within that system. And it's super useful because, you know, if you're looking for an endangered or very rare species, you know, they're really hard to find. But if you can just take a water sample and get the DNA, then you can confirm that that species is in that system. You just couldn't find it. Likewise, you can use it to identify invasive species before they get completely out of hand. And so it's mostly used in aquatic applications, but there are researchers that have been able to isolate eDNA from air. It's crazy. This technology is evolving so fast. It's fascinating.
Aren't they using that at the biological station to check for mollusks?
I believe they are, yeah. My favorite application of this was when they tried to find Nessie. I don't know if anybody heard about that. In 2019, they tried to find the Loch Ness Monster, which is really interesting because in order to identify a DNA sequence of an animal present within an ecosystem, like you need to have at least one sample of that DNA sequence, and I don't think so I honestly have no idea how they did it. I think the only thing that came out of that study was they determined that there are a lot of eels living in Loch Ness. So, yeah. Any other questions? Yeah.
Do you know the mechanism that necrotic flesh eating, because I know there's a lot of different bugs that eat dead animals. And we had a cat die while we were on vacation in our basement.
Oh, fun.
And I cleaned it up, and there were like some beetles and things that I've seen eat dead things outdoors. I've never seen them in my house, yet there they were with the dead cat.
So bugs have a really interesting way of getting into spaces that you just wouldn't anticipate. And before like the process of metamorphosis was described, so that's, you go from egg to larva to pupa to adult. There was a thing, I think it was called spontaneous generation, because flies would just show up in these spaces that they were like, there weren't flies here before, how did they get here? But they had no idea what the life cycle of these animals looked like, and so they thought that they just like sprang from the dirt. But yeah, they're really, you'd be surprised at where these animals can get and where they can lay their eggs without you knowing.
Do they smell like,
I do believe that there's like chemical signals, like not pheromones, but yeah, they can sniff it out pretty good, yeah.
I mean, I guess I was gonna go on that.
Okay.
Is there like a radius of?
I'm sure there is. They are, they're surprisingly efficient at sniffing things out, but it probably depends on the species would be my answer. Yeah. Any other questions? Okay. Thank you guys.
Thanks so much, Brenna. That was great. And thank you all for coming this evening. We will kick things off again in late September. So early September, keep an eye out for the next Bugs and Brews. Thanks a lot.